All right, let's talk a little bit about DNA replication. I'll skip through a few of these first slides because I know you don't need that information. You already know my contact info, so let's get past that. Let's get right to the bones of this thing. All right, so DNA replication. I'm about to throw a whole bunch of steps at you, and you're probably going to be wondering how you're supposed to learn all of these steps. So I'm just going to start by giving you a little advice on that. In this presentation, I'm going to cover as much as I possibly can about replication, but you really need to go over this information again and again. Make sure you read your textbook, you consult any handouts or lectures that your teacher might give to you, and make sure that you talk with your teacher if you have any questions. You can also consult, these are my favorite YouTube channels. We've got Amoeba Sisters Crash Course in Bozeman Science. They all offer some great videos on DNA replication that you can consult if you needed to. You just type in DNA replication and then Amoeba Sisters or whatever to find their video. So I highly recommend that because it's making it visual also helps. As I go through this presentation, I'll switch back and forth between my whiteboard and the presentation. So if you're looking for pictures, they are going to come. Just bear with me a little bit. Let's talk about what replication means. So what is occurring during replication is that we're replicating the double helix, the DNA molecule that's a double helix. We're going to copy that and make a new one. This is going to require many steps and many proteins. Replication is needed before the cell divides. We need an exact copy before we go through mitosis, for example. If there's any errors in the new copy of DNA that we're about to make, terrible things could happen. And the classic example of a terrible thing happening would be cancer. Replication is going to occur during the S phase of the cell cycle. So remember, during interphase, there's S phase, S for synthesis, which means replication, which is nice and confusing. Synthesis of DNA is the same thing as replication of DNA. It means the same thing. The first thing I really want you to know about replication too is that replication is called semi-conservative. That's going to mean that we're going to save one of the old strands of DNA and use it as a template to build a new strand. Let me show that to you. So let's just say we have a double helix molecule just hanging out, doing its thing. Here it is. There's a double helix. But we're in the S phase of interphase. We're about to replicate this bad boy. So how that is going to occur is going to require semi-conservative replication. So I drew my DNA molecule as red here to show you what I mean. So let's pretend that we pulled this apart, this molecule, and we're building a new one. So now I have two individual strands from this piece of DNA, and this isn't exactly how this works. I'm just giving you a really basic outline right now. We pulled this double helix apart, and here's one part of it, and here's one part of it. These red strands are part of the original molecule. Now I'm going to build a new molecule like this. So that one's a little wonky, doesn't matter. So I have two new molecules, but the red strand is from the old one. So that makes it semi-conservative. We use part of the old strand to build a new strand. So one strand is new and one strand is old. That's semi-conservative conservation here. Okay, so let's get down to what I have on this slide here. So I always recommend to students when you're doing anything with DNA, the first thing you need to learn is the basics. These are the basic steps of replication. You should memorize these basic steps in this order. And then later on, you should learn what happens in detail in each step. These steps are going to serve as your outline. So if you are learning replication, you start getting confused, I want you to be asking yourself, what step am I in? And then I want you to ask yourself, what proteins are being used during that step? We'll go into that in just a minute, but first let's just talk about this. The three basic steps. They're unwinding, complementary base pairing, and joining. 1, 2, and 3. We're going to go over these steps in a little bit more detail. I just want you to get these really hammered down in your head. Now, if all you could remember when you get to your exam is these three basic steps and the 5 proteins that I'm going to tell you about in the next slide, if that's all you could remember, you could probably explain replication and get, a pretty decent score on your exam. I'm not saying that's all you should learn about replication, but it's a really great place for you to start. So I talked about the fact that there's 5 proteins involved in replication. Here's a list of the 5 proteins. I would like you again to learn this list in this order. This basically covers the way that these proteins are going to be used, like the order that they're used in. There's some exceptions to that rule, of course, but I really know that from years of teaching students that this process, that organizing the information in this way will help you develop a basic understanding. I'm going to define what they are better later on, but we're keeping it simple for now. You're gonna know if we're talking about an enzyme, proteins are types of enzymes, right? So we're going to know if it's an enzyme because it will say A's at the end. And if it's not an enzyme, it's just a regular protein, it's not going to say A's at the end. So that's a little bit of a giveaway. So the first one is helicase. Number 2 is single-stranded binding protein. Number 3 is DNA primase. And number 4 is DNA polymerase, which is actually used several times for several reasons. And number 5 is DNA ligase. So remember those basic steps, and we're going to dive into them a little bit more at this stage. So let's start by adding some details to that basic outline. Step 1 of replication is called unwinding. You might suspect that we're unwinding the double helix. Since replication is semi-conservative, we need to access the template strand, which is the old strand. The template strand is the original strand from the molecule. So we need to access the template strand so we can pull the double helix apart. As you study these steps, make sure you're asking yourself what protein is used during that step and what the function of that protein is. So step 1 of replication, unwinding, is going to use 2 proteins. Those proteins are helicase. Helicase is an enzyme, has A's at the end. That's the giveaway. And it's going to use single-stranded binding protein as well. So what does helicase do? Helicase is an enzyme that unwinds DNA. It's super handy that the word heli is in there. You see it, heli, right here. Super handy that the word heli is in there. And that should hint you into the fact that it's doing something to the double helix. Helix, heli, is an enzyme. So that's a little bit of a hint there for you. So helicase is actually going to separate the double-stranded DNA that we start with into single strands so that they can be copied. So what is a single-stranded binding protein going to do? That is going to bind to the single strands of DNA that we unwound with helicase to prevent it from reforming the double helix. The chemistry of the double helix means it's always trying to stick back together. So if you unwind it, we need to have a way to prevent that from happening. Let me give you an image. All right, give that a second to turn on. There it goes. All right, so we've got a double helix here. There we go. Whoops, wonky, doesn't matter. So this is my unwound portion here. Sorry, it's blurry. So this is my unwound portion here, helicase. We'll just pretend that's helicase, right? Helicase is responsible for unwinding the double helix. Oops, sorry, that's still blurry. Helicase is responsible for unwinding this double helix. And then single-stranded binding protein is going to come along and bind to the unwound bits of the double helix to make sure it doesn't go back together again. Because we need this exposed so we can copy it. And then I just want to give you a little piece of vocabulary. I drew this a little wonky, sorry, but up in here, this is called a replication fork. It's just the location where the parental strands are separating. So that's the replication fork. All right, let's do a little quick reminder about DNA that you probably haven't thought about since like chapter 2 or 3, which feels like a lifetime ago at this stage. Back in like the chemistry of the cell, I think the chapter is called, you learned about complementary base pairings. DNA is, the chemistry of DNA, it means that it's made up of these nucleotide bases. And these nucleotide bases, they connect to each other, they bond together in the same way every single time, no matter what. So guanine always binds to cytosine and adenine always bonds to thymine. That should be what I mean. We have a double helix like this. So there's a guanine here and a cytosine here. There's an adenine here and a thymine here. They always hook up in this way, right? Or it could be the other way around. G, C here, G here, T here, A here. But those guys are always binding to each other. The other thing to know about it is that these strands, I should group that to begin with, always run in a certain way. So they call that 5' to 3'. And then the other strand will run in the other direction, which is 3' to 5'. What these numbers are referring to is the fact that in a DNA molecule, we number locations, which you don't have to know about too much for this class, thank goodness. But we always find the same thing at 5', and we always find the same type of thing at 3'. At 5', there's going to be a phosphate. And then at 3', there's going to be a sugar. So a DNA molecule has all of those components in there, which is what makes the two strands stick together. So the GC and AT are referring to bases that always stick together in the same way, and 5' and 3' are talking about phosphate and sugar that always stick together in a certain way as well. You really need to make sure that you know G to C, A to T, which is usually what teachers refer to it as, guanine to cytosine, adenine to thymine. They'll just say G to C, A to T. A lot of times teachers in this exam are going to throw a question at you, which may be like, match up the DNA strands. And the question is going to be like this. And I'm just making this up and I'm going to keep it simple because for the sake of the length of the video. They'll give you a string of letters and they'll say, this is a pretend strand of DNA. Can you match up the complementary base pairs? Well, we know that G always matches with C, and we know that A always matches with T, so we should be able to match this up pretty easily. G to C, C is going to match with a G, A is going to match with a T is going to match with an A. The string that your teacher gives you will probably be a lot longer, but I just want to make sure you're on the lookout for it. All right, so proteins used during this step. We're talking about step 2 of replication, which is complementary base pairing. So the proteins used during this step are both enzymes. We've got DNA primase and DNA polymerase. DNA primase is going to synthesize short RNA primers before replication begins. These little primers that DNA primase is going to put onto our unwound double helix is going to say, basically, copy this area, lay down bases here. And then we're going to use DNA polymerase. DNA is polymerase is kind of the superstar of replication. It does a lot during DNA replication. It does so much so that I give it its own slide. Here it is. So DNA replication. DNA replication, excuse me, DNA polymerase. Let's talk about DNA polymerase, which is used during replication. DNA polymerase recognizes the primers that were laid down by DNA primase and begins DNA synthesis at that location. It lays down complementary base pairs. It connects new nucleotides together. It proofreads the strand and it corrects any mistakes. And DNA polymerase will remove those mistakes by reversing direction and removing the nucleotides that are wrong. It will then change direction and resume making DNA. So it will help fix the problem and then start over again. When it comes to complementary base pairs, we have to talk a little bit about these things called Okazaki fragments. I'm gonna define that for you right here. Here's the word, Okazaki fragments. Okazaki is, I think he's Japanese, is a scientist from Japan. So that's where the name comes from. But we're gonna get there in a minute. So Okazaki fragments still has to do with step 2 of replication, complementary base pairing. Let's just talk a little bit more about the DNA molecule. So, and I will give you a picture for this. Just bear with me a little bit. So because DNA is always read in the 5' to 3' direction. So remember, there's a phosphate at 5' and there's a sugar at 3'. DNA is always read in 5' to 3'. The problem is that one strand of DNA runs 5' to 3', and the other strand of DNA runs 3' to 5'. That's called anti-parallel. The strands are anti-parallel to each other, so that's going to cause some problems for DNA polymerase that can only read in 5' to 3'. I'll show you that image in just a minute. Actually, we'll just do it now. Okay. So we have a DNA double helix. Like that. 5', 3', and then the other one is 3', 5'. Okay, so what we're gonna have here during complementary base pairing, there's a helicase here, unwound our DNA. And then we have these two separate strands, and DNA polymerase is going to start copying these strands for us. So we're going to have what is known as a leading strand and a lagging strand. The leading strand here is set 5' to 3'. And then you can see the 3' to 5', which is anti-parallel to the leading strand. It's running in the opposite direction is what that means. So DNA polymerase has no issue whatsoever with the strand that runs 5' to 3'. It comes along, it says, here I am, I'm DNA polymerase. I'm just going to call it DNAP. And I'm just going to start laying down nucleotide bases here. It's going to be great. We're going to make a beautiful double helix. It only has to do it once, and off it goes. So that's called the leading strand. On the other side, we have an anti-parallel strand to the leading strand. It goes in the opposite direction. That's a huge problem for DNA polymerase because it can't read in the 3' to 3' to 5' direction. It can only read the other way. So this strand is going to be called the lagging strand. Lag. You may have heard that like on the internet. If you're lagging, you're going slow, you're running behind. So what DNA polymerase has to do on the lagging strand is it has to be laid down many times on this. And it will make very, very short sections of nucleotides that are running in the right direction. But that's really slow to lay those down a little piece at a time. So the leading strand is always going ahead of the lagging strand. The lagging strand will always be behind. It'll always be taking longer to do replication. Now that brings us to our point about this Okazaki guy. The lagging strand contains these little fragments. You see, I kind of just drew them as lines here, right here. It contains these little bits of DNA that aren't quite connected yet. These little fragments are called Okazaki fragments. That guy Okazaki discovered them, so they got named after him. So the lagging strand contains Okazaki fragments. The leading strand does not. So yeah, there he is again, Okazaki fragments right there. All right, let's talk about step 3 of replication. So step 3 of replication is called joining. Let's talk about the proteins that are used. They're enzymes and they're used in this order. So first we have DNA polymerase. Yes, DNA polymerase yet again. Remember I told you DNA polymerase is like the superstar of replication. So what's DNA polymerase doing in the joining step? It's converting the RNA sequences that were laid down by DNA primase. Remember, we laid down those primers. We've got to get rid of those DNA primers because we're not going to have a complete DNA molecule if the primers are there. They're not a part of DNA. So those are going to be we're getting rid of that. And we're making sure that the primers are gone so we can convert a molecule into a proper DNA molecule. And then the second enzyme that we're going to use is called DNA ligase. So to ligate means to join right here. Join or glue, that's the definition of the word ligate. So that should give you a hint about what this enzyme is going to do. DNA ligase covalently attaches those Okazaki fragments on the lagging strand. Those need to be like glued together. We don't want those fragments in there. So ligase is going to glue together those fragments that were on the lagging strand. So what's the end result of joining? The end result of joining is 2 double helix molecules that are identical to each other and the original molecule. Each double helix is going to be composed of an old strand and a new strand. Let me show you what I mean. Okay. So double helix. Okay, we're in the S phase of interphase, so we're going to use that. to create two double helix molecules. Those reds are the olds, blue is the new. So the end result of replication is two double helix molecules. Each double helix molecule will have an old strand and a new strand. And voila, we finished.